Hello and welcome back. In this video, we will continue our exploration of epigenetics. Specifically, we will discuss whether Mendelian randomization is an appropriate causal inference technique in the epigenetic context. Well, you might think you're offering a whole course on this, so probably the answer is yes, but that is the simple answer. And in epidemiology, particularly in causal inference, nothing is ever that simple. The goal for us today would be to explore under which conditions it is, in fact, a good fit. Let's first remind ourselves why we're so preoccupied with causality in epigenetics in the first place. Here's a slide from my very first video of this section to refresh your memory. Essentially, the point here is that compared to GWAS or other studies of the DNA sequence, the causal relationships between the epigenome, which is dynamic, and various phenotypes are much harder to establish for reasons that are listed here on this slide. So if we do an epigenetic study and we find a statistically significant association between an epigenetic marker, for example, methylation of a specific genomic region, and a health trait, we cannot simply assume that it's causal. We need another approach. In this course, you already learned a good bit about one such approach, and of course, that's Mendelian randomization. It has become very popular in epidemiologic causal inference because of its elegance and its simplicity. It is relatively easy to implement, but as Steve already told you, interpretation can be a lot more challenging. The question that we'll explore today is, are epigenetic studies appropriate for Mendelian randomization analyses? What are the upsides and the downsides? I guess I started with the downsides. And what the right research questions are that involve epigenetics and can be answered with that approach. The very first paper that even mentioned epigenetics in Mendelian randomization in the same sentence, and that paper is cited here on this slide, actually presented epigenetic changes not as exposure or the outcome, but as a boogeyman, as a threat to validity of Mendelian randomization studies. To Obuanu and others, the authors of this paper, epigenetics was actually a really bad thing for causal inference. Let's explore why. You may recall from Stephen's very first lecture that Mendelian randomization is similar to a randomized trial. Randomization ensures that on average there is no relationship between various potential confounders and the treatment or the intermediate phenotype, and so you can estimate the causal effect of the instrumental variable on the outcome. And you can see that paradigm here on this slide. But here's the caveat. Some of you who've done clinical trials before may be familiar with this concept of intent to treat analysis. Sometimes that's known as once randomized, always analyzed. That means we're estimating the effects of treatment assignment rather than the actual treatment that the person got. And that might seem a little counterintuitive, but that's the only way we can get an unbiased estimate. Why? Well, because if we look at the treatment that the person actually got, as opposed to the treatment that they were randomized to get, the compliance with the treatment itself may be influenced by other factors that would induce bias. In a properly blinded study, it's actually not much of an issue, but it turns out that a similar concern may also exist in the Mendelian randomization paradigm. And it's all because of epigenetic effects. On this slide, it's circled in red, and you can see that it enters that graph in the same place as differential noncompliance on the clinical trial side, on the randomized trial side. If we have truly heritable epigenetic changes impacting gene expression, Obuanu and their co-authors showed in a simulated data set that it would impact the validity of a Mendelian randomization study in a very similar way to the manner in which noncompliance impacts the validity of intent to treat analysis in a clinical trial, in a randomized clinical trial. That's really the key word here. Let me tell you a little bit more about how that would work. The intent to treat effect which here is called beta ITT, and in Stephen's lectures you might know it as beta Y, can be written as the product of the effect of the genotype on gene expression, and in Stephen's slides that's called beta X, and the effect of the instrumental variable, which Stephen calls theta, and this paper calls beta IV. If this relationship is not exactly clear to you, please go back and listen to Stephen's second video, in particular the section on the ratio method, because that's what we're relying on here. But assuming you are comfortable with the ratio method and with this relationship, let's take a look at this particular causal diagram. Here we're showing the causal relationships between offspring genotype, an intermediate variable, which in this case is gene expression, and some outcome. The bottom part is just the usual Mendelian randomization diagram, and the only addition here is on the top, and that comes from the mother of this individual. What the simulation presented in this paper showed is that if you additionally introduce an environmental factor, let's say smoking, into the mother generation, and the mother's genes are not only epigenetically modified by smoking, but also these modifications are transmitted to the child, this ratio has to be modified to include that inherited epigenetic effect. If you're interested in the technical derivation of this relationship, I would refer you to the link to the paper on slide 4. But here, I just want to underscore that it is only a problem, and by problem I really mean bias, if and only if these epigenetic changes are inherited from the parent to the offspring, in this case, from the mother to the offspring. Here is the example presented by Obuano using simulated data. This example uses a SNP in IL-13, which is a gene that encodes an inflammatory marker, as an instrumental variable to look at the relationship between the IL-13 protein and asthma in the offspring. We also know in that scenario that maternal tobacco smoking during adolescence in particular results in epigenetic changes, and we assume that the child can inherit such changes. So at conception, the child inherits both the IL-13 SNP allele that would either confer higher or lower inflammation, and the epigenetic alterations conferred by mother's smoking. Among non-smokers, and that's what you can see here, we really do not have to worry about anything epigenetic, and the resulting Mendelian randomization estimate is not biased, because there was no inherited effect in addition to the effect of the allele itself. But in the offspring of the smokers, we see a spurious association between smoking and the expression of IL-13 because it includes effects of the allele and of the epigenetic changes. So we have an association here between the instrumental variable and the confounder, and that goes against one of the Mendelian randomization assumptions, and it biases the estimate. Just how much does it bias the estimate? In this particular simulation, the authors assumed that if the epigenetic effect is positive, and in that case, excluding it from the analysis results in a gross overestimate of the effect of the instrumental variable. Again, if you're interested in the technical side of things, I would refer you to the paper that's linked earlier, but here you can appreciate that the bias due to not accounting for epigenetic effects was quite substantial. Now here's my second trick question for you. Why do you think they used simulated data as opposed to real data? Because look, it would have been easy enough to show in an actual epidemiologic study. So why go through this labor-intensive mathematical exercise of simulating an entire data set? Well, it's because the real data set does not exist. You may recall me emphasizing in the previous slides that this epigenetic bias would only be a problem if the epigenetic effect is passed down from the parent to the offspring. There are no real human epigenetic data, at least when it comes to methylation, where this happens. So they had to simulate. This diagram from the previous module reminds you that in the case of DNA methylation, we really do not have to worry about transgenerational inheritance. That is not a thing. And it is because during very early embryonic development, we undergo 2 cycles of very active demethylation, and that erases almost all of the parental epigenetic marks. That may or may not be the case for other epigenetic mechanisms, but in the current state of technology, most epigenetic data you'd be working with is restricted to methylation. And that's good news for you, because that means you will not have to worry about that bias that Obuano warned you about. Now, I included it anyway because it's a worthy discussion to have. If at some point in the future, evidence for transgenerational inheritance of other epigenetic markers is generated, you would really need to be mindful of this bias. With methylation, not so much. So let's recap what we discussed so far. I showed you a purely theoretical scenario where epigenetic effects can bias Mendelian randomization estimates, and then we realized that it's not going to be a problem in most studies of DNA methylation because it is not usually inherited. So we went from epigenetics being a big problem to being kind of irrelevant. The next question is, are there scenarios when using epigenetic data in Mendelian randomization studies is actually a good thing? And of course, like in many other situations, it depends on your research question. When we treat epigenetic effects not as some nuisance variable, but rather as an exposure or as an outcome in their own right, well, then things start to look up. What do I mean by that? Imagine we observe a statistically significant association between methylation in a specific genomic region and, let's say, BMI, body mass index. It is just an association, and we know better than to call it causal, so we decide to investigate it further using this Mendelian randomization approach. This graph is probably familiar to you from Stephen's lectures. So in our example, we can think of this observed methylation pattern as the exposure and obesity as the outcome, or BMI as the outcome. And we can interrogate that association using an instrumental variable that captures methylation. Later, we'll talk about what those variables can be. Alternatively, we know epigenetic patterns are dynamic. And so we can think, well, what if it's not methylation that's causing obesity, but instead the epigenetic changes are downstream of this phenotype, so obesity causing methylation? And in the case of obesity specifically, we actually have evidence that that's the case. Well, in this scenario, you can choose a different instrumental variable, one that captures obesity rather than methylation, and conduct a classical instrumental variable analysis. That is the basic framework for bidirectional Mendelian randomization. Before we get there, I do have some good news for you. One really nice thing about epigenetic changes, and DNA methylation in particular, is that they often have very strong instrumental variables that are not hard to find. Recall that that's our first assumption of Mendelian randomization, a strong relationship between a variable and its genetic proxy. I will tell you that it does not get much stronger than the relationship between SNPs and CpG site methylation. In some of our studies, we've seen p-values in the 10 to the minus 200 range. These SNPs are called methylation quantitative trait loci, or MEQTL. Some MEQTL are easier to find than others, and your statistical power to do so may depend on different things: SNP allele frequency, the variability in the methylation status of your locus. That could differ across stages of development or tissue types, and linkage disequilibrium structure, which is sometimes different across ancestry traits. So all those things could impact your power to find proper variables for your methylation. There is a really large body of evidence documenting all kinds of such variables, or MEQTL, and they're summarized here in this graph. Usually, the physically closer your instrumental variable is to the site of epigenetic variation in the genome, the more robust your associations will be. In scenario A, you see a local effect where the SNP and the methylation site are within 1 megabase of each other. That's really what you want for a good instrumental variable. All other effects are considered distant, and that could be on the same chromosome, like in panel B, or on another chromosome, like in panel C. Because there are many millions of methylated sites in the genome, local SNPs with respect to one methylation site would be distant SNPs with respect to the vast majority of others. In D, you have a situation where the SNP actually overlaps with the epigenetic site, and those are called CpG SNPs. You should probably remove those from your data set to avoid spurious associations. Other SNPs in linkage disequilibrium with the CpG SNP would also be associated with that effect on methylation, so that would also fall under panel D. In general, your best bet would be a local SNP that is not on the methylation probe itself. Such SNPs, sometimes we call them cis MEQTL, often explain a high percentage of variance in the methylation site and thus fulfill at least the first assumption of Mendelian randomization analysis. Another piece of good news is that you do not have to be confined to one SNP as an MEQTL. So just like what Steve showed you, you can combine multiple MEQTL variables in your approach, and you already know the advantages of that. For example, you know that using multiple instruments ensures you against the possibility that any one of your instruments would yield unreliable estimates. However, even if you have these wonderful, robustly associated MEQTL instruments, you would still need to check that your other two assumptions are fulfilled. And that means independence of your instrument of any potential confounders and also lack of pleiotropic effects. If you use multiple MEQTL SNPs, pleiotropy in particular deserves a very careful look because your MEQTL can influence all kinds of biological pathways, and you can use standard methods like MR-EGR to test for that, just like in a typical garden variety Mendelian randomization analysis. Another important thing to know about MEQTL is that they're tissue specific. Your SNPs are the same in all tissues, but your methylation patterns may not be the same. So just because a certain SNP is strongly associated with a methylation locus in whole blood does not mean that that same SNP would be associated with methylation in the same locus, for example, in the brain. There have been studies investigating just how different those associations may be, and they've produced a mixed bag of evidence. On this slide, I have linked two good papers that have explored that question for those who are interested in the details of cross-tissue MEQTL associations. The take-home message from this part of the video is that assuming you have the right tissue, and assuming that your other two assumptions check out, and that's no pleiotropy, no associations with confounders, well, if all of that works out, MEQTL are actually pretty good instrumental variables for methylation. You can usually find them in the same genomic region as your epigenetic marker of interest by simply regression your methylation proportion on all possible independent SNPs, and then either use all of them or choose those that are most likely to satisfy your assumptions. In future videos, we will see examples of such variable selection. So now let's go back to our favorite slide, which might as well be titled, "What's Wrong with Epigenetics?" Well, we have these challenges in epigenetic studies that preclude us from simply assuming that any observed association between an epigenetic markers and a phenotype is causal. If you come from the genetic world, it's a bummer. If you come from the traditional environmental factor epidemiology, well, that's really not news to you. You're used to that scenario. So today, we explored how we might begin using Mendelian randomization to address some of these causal challenges. Just like a randomized trial, Mendelian randomization, if done correctly, is pretty good at addressing this third issue of bias due to confounding because if your assumptions are satisfied, we can assume that our confounders are on average equally distributed across genotype groups. Tissue specificity is still a concern, and so is time because we still do not know what came first, the epigenetic mark or the disease phenotype. And that is what we'll cover in detail in the next video. But before we get into this chicken and the egg question, let's sum up what we've learned so far. Despite what these early simulations showed, in the case of DNA methylation, we do not have strong reasons to worry about epigenetic changes threatening somehow the validity of our estimates. And that's because they're simply not likely to be inherited. So instead of treating them as a bad thing, instead we can use epigenetic markers as variables of interest that help us understand the underlying biology. And we know it can be used either as an exposure or as an outcome. In both cases, Mendelian randomization can be applied to support causal inference. Today, we also talked about selecting instrumental variables for DNA methylation, and those are called MEQTL, and we've set the stage for bidirectional analyses that we will cover next. Thank you.